All right. Interestingly, I presented this as the first lecture last year, and this time I decided to present as lecture 10. And again, I must have reiterated many times that it's more of a cramming material, and I'm not going to go through those slides which have that cramming material, but I can just give you an upshot. And as a matter of fact, while I'm talking to you, I read this new chapter as well, and it's very interesting, and it's like changing the whole concept. So I'm gonna spend today's 50 minutes on the concept, and then you can do the remaining part, and you will have an idea why and how and things of that nature. When we say commensal bacteria, and commensal, this is basically I think a restaurant in Toronto or Montreal or something like that. So by definition, I always struggle with that. By definition, commensal means that one who eat at the same table. That's what commensal basically means. I thought it was an English word, but somebody told me it's a French word, so I'm not an expert in French, but whatever. So I'm going to spend some time on giving you an idea that how do we deal with these commensal bacteria, and they are very important in terms of one of the top journals. You can see Nature Medicine will have the front cover in 2009 on a review for commensal bacteria. And I myself, being a microbiologist, I'm very much interested in that, and that's going to change the whole concept of medicine. And I must have told you many times, those students who went with me for ASM and AAI last year, is everything is now turned towards bacteria, and I'm going to explain to you why and where do we go from there. So today we'll talk about normal flora and what are the factors that affect that. I'm going to talk about some concept in terms of definition. I'll also talk about some of the other things, but the most important thing is that these bacteria happen to be at different places, and there's a set number of bacteria, there's a set names of bacteria, and I want you to remember some of the important ones. Let me put it this way. If you look at a particular place like oral flora or nasal flora or oral flora, ear, or different flora, it's good for you to know what are the culprits in terms of normal as compared to abnormal. That's the whole idea. Okay? And then in the end, there are, like, I think 10, 20 slides just quizzing you and asking you some of the questions you need to know. Now, the whole concept which has changed for the last couple of years is coming from technology, it's coming from biotechnology. Government decided out of nowhere that let's fund a project which is called Human Microbiome Project. And remember, they already funded Human Genome Project. And we talked about the changes in medicine which are coming up, evolving in terms of medicine, personalized medicine. So that's another frontier which will change, evolve the whole medicine to such an extent that even schools like Harvard and Yale and Johns Hopkins and so on and so forth, they're changing the whole curriculum. And we ourselves are going through that process. And those of you who are in the curriculum committee, they would know that we're talking about the CAPE 2016, 2013 now, and 2016. Anyway, so since we have technology, we have a way, so why not do it? The idea was that we used to ask students to remember the names and culture it, but since we have molecular and genetic techniques, why don't we use that genomic techniques and pretty much use the computer, get all those DNAs from there and feed it to the computer. Easy thing. That's what the project was. And then the idea at that time was that we wanted to establish, we call microbial communities. So we thought that let's pick up specific communities which basically are responsible for quite a few diseases. And very rightly so, if you remember, I said all those opening into our body are the places where bacteria would or microorganisms would try to penetrate or go in. And that's the challenge we have. That's something that we share with the atmosphere. Whether we like it or not, it's something that we share. And we'll also give an idea what are the bacteria that are good for us and what are the bacteria that are bad for us, foes and friends. It's just like listing of bacteria. And then again, what they did at that time was the researchers, they picked up, of course, recommendation by the microbiologists and immunologists, and simple for you to pick up mucous membranes, skin and mucous membranes, and pick up specific sites, for example, nasal passages. Everybody gets flu, everybody gets sick, pneumonia, so on and so forth, sore throat. Look at oral cavity and look at gastrointestinal tract and look at urethra and vaginal tract. So these are some of the things that they said, okay, these are the places that we have a problem and we have bacteria sitting over there. Let's go for them and kind of figure out what's happening there. A lot of work was done, and I'm not going to share each one of things, but I'm just going to share you some of the things that you need to keep in mind, that if when we did the genome of human cells, so we were thinking maybe something to the order 10 to the 13, as compared to when we started looking at microbial genome, microbial cells, we had in the order of 10 to the 14. So for every one cell of human body, we have 10 bacteria or viruses or whatnot. You can imagine that it's like a philosophical discussion as well. Are we human or are we microbe? Because we have more microbes as compared to that. And then again, there are many factors that would affect that community. And again, I'm going to discuss that. And I'm pretty sure many of you in consultation to the patients will come and be asked about this question. People out there are very much worried about many things. So you have to keep in mind that diet, antibiotics, your personal hygienes, where do you live, whether you live in village or urbanization, and family size and so on and so forth. All these play an important role in terms of what really happens. Okay, let me see if I can enlarge that. I usually struggle with it. I'm sorry for that, but I don't know how to do that. Is it a little bit better, I believe, right? Yeah, but it doesn't allow me to change. Yeah, that's better for me. Okay. Now, what I want you to keep in mind is that we got friends and foes. So the friends who eat together on the plate, or that's what it's a commensal. We call them commensal bacteria over here, as compared to the pathogens that are there to cause problems. Now, also keep in mind that I'm not going to go again into details of host response. You pretty much know we have a very patent immune system at all mucosal and skin surfaces. That's for sure, right? And then again, all those cells are important. The most important thing you have to keep in mind is that when I was about to say most of us, but it should be all of us, were in the womb of the mother, so we were sterile, unless there's a test tube baby. Anyway, but that again has to go into the womb. All were sterile, absolutely no microorganisms. So there was a time, 9 months, the moment the baby is born, exposed to the microbial world, right? And then the fight goes on from the day you were born to the day you die. And you know who wins? Microbial wins. We're going to chew you up, eat you up, decompose you. Anyway, then again, unless you get, I don't want to go in that detail, but anyway. So I think you got a message about sterility. So we have microbiome. All of us have a microbiome. Trillions of bacteria, fungi, and viruses. They are all there, and they all together are on our bodies. They are together having genes. That's the important thing. Like our cells have genes, they have genes, right? And we put them together, we say, this is my human microbe. Right now, you don't know. If I was to ask you, what's your human microbe? Nobody, so your physicians would not know. But they are going to a technology where each one of us would have our genome of our cells, and we all will have a human microbiome done for everything. Right now, it's still, it's been done, but it's still in the process. Now, also keep in mind, when I say your human microbiome, keep in mind, just like babies, where we were still all together, our body itself, which is the real body itself, like tissues, still are sterile, like blood, like kidneys. You name an organ, they have to be sterile. If they are not sterile, then we have a problem. The only exception is that your immune system may be hiding some of those bacteria, viruses, and fungi, intracellular parasite that you are not aware of. Or your organs become diseased and you have a problem. But today I'm going to deal with, you can see oral cavity, nasal passages, lung, skin, urogenital tract, and GI tract. As a rule, anything that opens to exterior, anything allows air contact or contact with the rest of the body, there have to be bacteria there. And that's what your human microbiome are. Now, when we go for their dissection in terms of number of bacteria, so as I said earlier, if I was to look at all the cells of a person, so this is our cells, right, in terms of human cells, and these are the bacterial cells that we are body, and these are the fungi. And again, it's just a research process. It may change. I'm telling you right now today, there are no viruses, but I may change next year depending upon the research. Okay, but right now it's for sure that we need to know what are the bacteria that basically all are friends. So you have to open up another Facebook group or whatever group and say, hey, buddies, if you know them, right? And you will have a problem because there are trillions of them. And so unfortunate that none of us, including, I know a little bit, we don't even know who they are. What are their names? We only find out when they cause problem, and we have to go to the doctor. But anyway, keep in mind that when we talk of all these things, if you were to take all the bacteria which are on each and every part of the body, right? And if you were to weigh them, they're about 2.5 pounds. It's pretty heavy, right? And then if you were to put them in a volume, so you can see they're going to have at least 3 pints of bacteria. There are a ton of them, tons of them. And you can imagine the problem we will have in terms of them causing disease. Now, I'm not going to make your life miserable unless you want to go into genetics or you want to go to human microbiome. But this is how they do the analysis. So you can see very complicated computerized thing, a map of diversity in the human microbiome. Each one of us, like our fingerprints, different from each other. We may have all different sort of bacteria. That's an idea that, as I said, new medicine is why do we need to give the same medicine to everybody? That's now a big challenge for the medical field. So they want to give you the medicine according to your gene. They want to give you medicine according to your microbiome. We are not there yet, but you can see from here how big a role genetics will play tomorrow. Okay, because this is how the whole direction of medicine is moving toward. And then again, it's going to be a mapping. So next time a physician, when he has a physician's office, he may open up a clinic, and he has to have a computer guy as well, an analyst as well, right? Because of the data. That's why they're saying right now the hardest job in US is analyst, because everywhere data has been generated. So they're looking for the data analysis. How are you? They need experts in that, right? And I'm not going to tell you how much is the pay of people who work on that. I'll just give one example. Like one of the CEOs of Google will get like $200 million a year. So you're looking at a big time money in terms of what is going to come up. But again, the question is very diverse. A map of human microbiome. Okay. Having said that, they still think the problem is, okay, let me tell you, the problem is like before we have these genetic techniques, we used to culture them. And you know, most of you were struggling even to gram stain the bacteria which are full loaded with bacteria. Just imagine if the patient was waiting for your gram stain to start initial antibiotic, where would that patient be by now? Right? So to make our life easy, we cannot culture them, let's be honest. Right? So we can still do their genome. So we always tend to go for technology as compared to manual thing. That's so real these days. I mean, you can be an old school, but that's the way it is. Since, and that's an argument for being going through those techniques, since it's estimated that small portion of microbes work, that's why we used to wonder if I asked you to take your nasal swab and culture it, you will grow 6, 7, 10, 15 bacteria. And that's what we wanted you to remember last year. But now I'm myself debating that if genetics were done on those sphere and there are 1 million bacteria, so why would we want to even waste time of culturing it? Why would we want to waste time in memorizing it? But the good thing is that still there are some known players as compared to unknown players. So we're going to go and play with the known players. Okay, so just to give an historical prospect, especially was started way back ago, and then again the Human Genome Project. You're not talking about microbiome. I'm talking about Human Genome Project. Your Gene. They started in 1990, and they pick up 3 billion nucleotides, and we now think that there are 23,000 proteins that encode our human DNA. So that's one part of the work. Keep that one part of the work where I said human versus microbes. Now, when they talk of the microbiome, when they talk of, for example, a microbiome project, there's a typo. It's not a genome, it's microbiome. There's a typo. I just typed this morning. So this was launched in 2007. So you can imagine pretty recently. And then again, they took the samples from all these places, 1, 2, 3, 4, 5, basically, from healthy adult volunteers. What they did was they sequenced them for the region of 16S ribosomal RNA gene. So that's what I'm going to ask you. Because that's important, that 16S ribosomal RNA gene, and then they want to determine the whole genome. So they determined the whole genome from different parts of the body. Now, what they found out, that number one, there was a lot of genetic diversity. The maximum genetic diversity of bacteria was in the intestine. And the least complex was vagina. So that kind of gave them an idea, because in terms of opening, in terms of mucosal surfaces, you can also look at the surface area that's exposed to the exterior. And you also look at the surface area which is there in terms of having normal population, for example, colon. So whatever trillions of microbes I told you, I'm not going to give you an exact number, but majority of them sit in colon. That's why the feces are nothing but bacteria. So all these things which are degraded in terms of our human consumption is nothing but bacteria. So what they did was, so they want to label that microenvironment in terms of mouth, gut, skin, vagina, and so on and so forth, and want to come up with a standard, I would say like a chip, because they're talking about computerized things. Now, then they came up with an idea that, each one of us may have a core microbiome. Each one of us may have a core microbiome. This means that they are always present at those specific sites in 95% or more of individuals. So that's a shared thing. If I tell you the Staph aureus is going to cause this thing, so for 95% of you, it will cause that, right? So we have shared microbes. So that's important. We call those microbiomes as core, okay? Because they are present in our mouth, nose, intestine, skin, and as we said, in females in vagina. But there are some other which may be there, but they are those which are in continuous transition, right? So that is where we play the game. That's where the diet has to play a role. Your hygiene has to play a role. Is your place of living has to play a role. Your physical interaction with other people, so on and so forth. Okay. But by far, this is called core. So secondary microbiome is that 5%, and that again will also be dependent upon if you are normal. And if your immune system is normal, you tend to have the core microbiome. Does that make sense? Right? Because your metabolism, your immune activity are pretty much standardized. But if there is a little bit of alteration in that, you tend to acquire, right, or play with those different variables that you may have. But they are called secondary microbiome. Okay? Secondary microbiome. Now, what they did in the human microbiome was, so they not only took those openings in our body as the major player, but the problem to them was skin. Because remember, many a time if you go in the 9 lectures that I told you, most of them were skin infections or mucosal infection. So skin is the widest area. It's the largest organ in the body, skin. So we tend to have different microbes at different part of the skin. So even if there is a crease, axilla, perineum, popliteal fossa, you name it, even if there is a bend, a crease, that's going to host a different set of bacteria. So you can see from here, we picked up quite a few bacteria, and we wanted to, even on plantar surface of feet, and we wanted to make sure and make sure that what are the overlaps and any similarities or dissimilarities. Okay. So of all the public places you have, which will be the best way for you to share all your, for most part, all your microbiomes with the community? What is the public place? I'm just asking you this question. If you were to go there and you can have a good kind of sharing of all your human microbiome with the rest of the community. Hospital? If you go to hospital. Anybody, any other guess? Louder. Swimming pool. So if you ever want to share your microbiome, just jump there. That's the best. You listen to the point? The thing is this, that because all your skin has all those things, that's why they keep on advising you, wash yourself before and after, but nobody knows, nobody, who's washing and whatever people are doing there, but that's the best way for you. That's why we usually have a discussion. I would very rarely see all those microbiologist immunologists jumping into the public pool because they are paranoid of all these things which are there. Anyway, the other important thing you have to keep in mind is that when we talk healthy microbes, or these one, because I'm trying to inculcate this idea, you cannot get rid of your friend. You should not. That make sense? There's no such a thing as, you say, a super hygiene. Basically, super hygiene is a problem. I must have told you in immunology. What's the problem with the super hygiene? There's a theory called hygiene theory. What's the problem with super hygiene if you were ever to maintain that? Because what they are saying is that, especially with those people who are from so-called underdeveloped, so they get a lot of infection because of lack of hygiene. So their immune system is fighting those infections, right? And busy with that. In the developed countries, they're more sanitary and more hygiene. So their immune system is free. It's not going to fight infection. So then they will be challenged with cancer. So they are thinking that that's the price you're paying because of your hygiene theory, that your system is going to go for all these autoimmune diseases, so on and so forth. But the point is, you have to have healthy bacteria because they help you to educate your immune system, especially on mucosal surfaces. So don't mess with them. Let it be. Now I'm going to tell you some important point where you draw a line. How far can you go? What is the real sanitation, right? And what is not? I mean, you can clean yourself after going to restroom, but you cannot clean your intestine. Correct? You see? So there's a limit, but we do prepare people when we do for colonic surgery in a different way. But again, keep that in mind. So you can see when you deplete your microbiota, and that's the problem with the use of antibiotics, because antibiotics would not care. Friends or foe, all will be eliminated. So that's why we try best not to give, antibiotics at all times. So you can see from here, if you give antibiotics, for example, you're going to eliminate all your friends, and you have depleted microbiota, and these are like your good friends over here, right? And they form a big layer, and like all seats are full. Somebody walks in, no seat for him. Okay, go out. So all these are good bacteria. I mean, just giving the example. And then the pathogens are waiting for a seat. Unless you vacate a seat, they're going to go in. So these are like pathogens. They come around, all house full, no seat. But if you give an antibiotic, you're going to deplete this layer over here. You can see from here. And you allow these pathogens to go in and kill your cells. So the idea is, if you're going to remove that layer by overzealous sanitation, by unwanted sanitation, right, and you tend to remove them, you give a chance for pathogenic bacteria, in this case, to jump in and kill your cell. That makes sense? Okay, keep that in mind, and I'm going to quiz you. Okay, now, as I said earlier, these microbiota can be influenced. Now that's where now you want to draw a line, right? And definitely you can write a question. I can take this question in the end. Personal hygiene is the major factor that's going to change that or alter that. Use of soap, of course. Deodorants. Okay, you're going to kill the smell. The smell is coming from bacteria. And unfortunately, even your normal friends may be smelly. But if you overuse your deodorants, you basically are killing them up. Okay? If you're going to take mouthwash every 5 minutes, I'm just joking, or mouthwash with antibiotics, you are playing with them. See? So that's another question that physicians you have to ask the physician, not look at the advertisement on TV. That can be misleading. Okay? Antibiotic or alcohol, is just like a very important antiseptic agent. Okay? People go for skin peels. We talked about that. I showed you the little tiny fish, chew up the outermost layer of the skin. People want to take enemas. There is a concept in some of the medicines that, hey, we need to cleanse your bowel because you are sick because you have not had this bowel cleansing. So the enemas are there for people. People induce diarrhea, and they think that's a remedy. And then there is another thing that's coming up. Let's get rid of your bacteria. They are causing you disease by giving you diarrhea or enema and give you good, healthy, friendly bacteria. So that's out there. I'm not going to discuss that right now. It's going to take me many hours to explain that to you, but I just want to give you an idea. All these things have to have a very solid concern. Does that make sense? Vaginal douches. Now, also keep in mind that very common in this country, very high rate. Women will clean themselves up pre and post sex, for example, pre and post menstrual cycle. But guess what? Not recommended by physicians. Okay, And the fact is because firstly, it's going to wash out your healthy bacteria, number one. Number two, all those gadgets which are available, which is going to squirt fluid into you, are going to push your normal vaginal flora into your cervix and cause disease. So I'll discuss that as a very common STD. It's called vaginosis. Very common cause for vagina. So these are some of the things which are out there. Of course, diet has to play a role. Water source, medicines, all have a role to play in terms of what is happening to a particular, person, in that ecosystem. So what I'm trying to do in this lecture is not going to go and take one side, but I'm going to give you a reasonable physiological opinion, right, about the concept of normal flora. Okay, and even if you were into that position, you can make your own judgment in terms of advising people. I mean, I'll just tell you one of the things that is coming these days is that two of the diseases they call inflammatory bowel disease, Crohn's disease, and ulcerative colitis, very severe, very high incidence. So they are thinking that they are caused because people lose the normal chronic commensal bacteria, right? So now what they're doing, one of the treatments, what they're doing is that those people who get it, they want to give them enema for some of the healthy chronic bacteria, right? So I'm just giving an idea. The other important thing, for example, in some of the latest, it's like a research work, not given in textbook, that they're also thinking that those babies which are born by normal vaginal delivery versus those born as a C-section, the C-section would not acquire mother's normal vaginal flora. So that's a handicap on that person to educate baby's immune system. So what they would do is, if that is the case for the C-section, they would rather pick up a normal vaginal delivery, vaginal flora from the mother and apply it to the newborn baby, even respond in a C-section. Gives you an importance of the normal commensals which are there, and you need to get exposed to it. That makes sense? So this is something that you have to keep in mind. And when we look at, for example, again, very complicated, I'll just give you a gist of it, but it's coming up. I mean, it's going to be a challenge for all the physicians, all the people like myself who are teaching, with all these technological advances that are coming up in terms of, for example, if you were to do a genetic survey of different places of the body as like A, and you can, and these genomes, remember, these genomes are real because you cannot change the DNA. So these genomes are real. So these genomes are done over here. And when you, for example, let's say, give an antibiotic, so you can see there's a change there. If I was to say that the red represent pathogenic bacteria, which are sure to cause diseases, and they are less as compared to, you have less foes than friends, and then you give antibiotic or any other treatment, or change the diet or anything, so you end up having them more. So there's a big change in terms of bacterial population, to such an extent that they are even saying today, I remember last year I went to American Society for Microbiology conference in New Orleans, and they were telling even diabetes, weight, hypertension, everything is coming from your chronic bacteria. But this is very trendy, very fashion these days, because they think, because these are the bacteria that educate your immune system, if you mess up, play with them, then again will cause a problem in terms of response. Okay? Now, rest of that was the added slide. Rest of the thing is more of a cramming thing that I'm just going to run through, just conceptual for you. As I said earlier, infection can come from any opening, can enter us from any opening, right? Clear? Infections can also come by injury, scratch, or also come from arthropods or any insect that can inject malaria, whatever, right? The other concept, remember when we talk in immunology, we talk about receptor shedding. The TNF receptor can be shed, placed, so on and so forth. Now, in this case, what happens is that you may be colonized with this bacteria. So you have this normal set of bacteria, right? And you get colonized with them, and you are shedding it. So you're washing, blowing, whatever you're doing, talking, you are shedding your bacteria. So that's what we call shedding. So that's a concept for some of the people. For example, if you are nasal carrier for Staph aureus, well, it's good for you, but you cannot be working in a hospital, or you should not be working in restaurants so that you can pass it on, sneeze on all the food. If the food is just brought in front of you, who knows what's happened, in the kitchen. Okay, so that's not a good thing. But as far as carriers is concerned, and as I said yesterday, women are asymptomatic for gonorrhea, and they can give it to their partners, and they have STD. And they themselves would never seek attention because they didn't have any problem. Okay, so these are some of the things that you have to keep in mind in terms of understanding the concept of disease and colonization. Now, if you draw a Venn diagram in terms of, let's say, for example, the benefit this bacteria give it to us versus harm, right? Or how much are we dependent on them, you will see, to the best of our ability, our relation with them is symbiosis. Right? Symbiosis. So the definition for that is that, for example, had it been parasitism means unilateral benefit. You make food, I eat, right? So I'm parasite on you. So they are there for their own benefit, like viruses. We don't really, as of now, get any benefit for getting viral infection. They get the benefit. So that's like unilateral benefit. They come and look for shelter and food, because when we eat, colonic bacteria are going to thrive on our food. They eat that. They metabolize carbohydrate. So they produce that. Okay. And mutualism, which kind of we would have kind of desired, would have a reciprocal benefit. But by the end of the day, it's a combination of all three we call symbiosis. And again, I think as we move along, you'll see that we usually would rather have them there. Most of the bacteria are there in intestine, right? All full of bacteria, and they are very specific bacteria. When we say normal commensal, and why do they say normal? These are some of the positive effects that we have. They participate in metabolism of food. They provide us essential growth factors. They protect us from highly virulent microorganisms. They stimulate our immune response. This is something that we pretty much have discussed many a time. The other concept is, other than the core microbiome, these are in continual state of flux. So they're changing as well. Depends upon age, diet, hormone. That's why I said, especially for, as I said yesterday, in terms of STDs, that depends upon the age, pre-pubertal, pubertal, postmenopausal, menopausal, depending upon estrogen, for example. So that will also play a major role in terms of what bacteria do they harbor. We already talked about that. The human fetus is perfectly protected. And then again, I always give you an example that once the baby is born, skin gets colonized first, followed by oropharynx and gastrointestinal tract, and so do the other mucosal surfaces. That's why physicians and gynecologists would encourage people to go for vaginal delivery in terms of being it normal, physiological, unless, of course, there are indications for that. So why do microbial population change? Well, they can change if you happen to go to hospital. They can also change if you are given antibiotics, right? And by far, most of the bacteria, for example, when we talked about nosocomial bacteria, and this is the term that I use, which are mostly are avirulent, right? and they are present in our oropharynx, like Klebsiella and Pseudomonas. They can invade the lungs and cause pneumonia. Don't confuse that with the concept of antibiotic resistance. So these bacteria can become pathogenic. Okay, we talked about Clostridium difficile. If you overtreat, you kill the good ones. And for normal bacteria, commensal bacteria to be normal is also in normal numbers. If you outgrow them, like Clostridium difficile, by all means, is normal flora. But if they outgrow in number, they cause damage. So also keep that in mind, okay? I already told you the difference between colonization and disease. Some bacteria can colonize us. They may be temporary, permanently, but we do care when they produce a disease. So you should know that you may have a colonization of a particular bacteria and may not get disease or infection. It's pretty much the same thing. When we say colonization, it can be transient, right? Hours and days, it can be permanent, but it doesn't really interfere your normal functions. But when we say disease, so disease is when a microbe, whether it be a commensal bacteria, whether it be a pathogenic bacteria, will try to cause tissue damage. So that's where, because organ damage, tissue damage will take away the normal function of that particular organ. If you get infection of the lungs, you will not be able to breathe. If you get infection of the liver, you will not be able to have normal function of the liver, so on and so forth. So this is where it is coming from. It could be because of bacterial toxins in terms of pathogenesis, but it could also be because of the acute immune response that we generate to eliminate them. Again, this is, I'm going to go through that, and this is now your take for memorization. Each one of them is a question for you. So the question will be, which of the following are strict pathogens? The definition is they always cause disease. They can never be your friends, regardless. You cannot say that they are commensal. And these are Mycobacterium tuberculosis, Neisseria gonorrhea, Francisella tularensis, Plasmodium, and rabies virus. They are microbes which are called strict pathogens, always cause disease. Some of the pathogens we call opportunistic pathogens. They will cause disease for most people, if they get an opportunity. They may be normal commensal, like Staph aureus, Escherichia coli, Candida albicans, right? They should not produce disease in normal setting, but if you introduce them into your system by cutting, abrasion, or any other means, they are going to go and cause infection. So they're opportunistic. So which of the following sites does not have normal flora? Blood, of course. Blood has to be sterile, but all others have a normal flora. Okay. Well, this is type of questions that should come from this lecture, because you need to know the normal bacteria for anterior urethra. For example, many times if you go for a urine test, they say get the midstream catch, because your urethra may have those skin-related, or you have to clean yourself, and then go for the voiding. But these are some of the things that you also have to keep in mind. Corynebacteria is not considered as a commensal bacteria, but lactobacilli and coagulase negative Staphylococcus are considered as normal population of anterior urethra. Now, we don't teach them to you a systemic microbiology. I usually have a workshop on systemic microbiology, but what we do is that we divide that into microorganism causing upper respiratory tract infection. So, but you do ask that, what are the organisms that are normally present in upper respiratory tract and cause infection? Most common are anaerobic. Most common anaerobic are these, and most common aerobic are these. So this is something, again, there are quite a few mnemonics for you to remember, but this is something that you need to remember, so you know when you're asked which aerobic bacteria can cause upper respiratory tract infection. That's called systemic microbiology. And there's nothing but your memorization. Okay, I'm going to let you read that. Some of the other important, since this is a journal lecture, there would be fungi or parasite as well that may be sitting over there in those particular respiratory tract and may cause problems. But we'll discuss them in detail as we do the other things. These are potentially pathogenic organs, and most of it are already there in the listing that I gave to the students. So I think if you are 90 students, so most of them that we need to cover in terms of bacteria, in terms of viruses, fungi are already there. That's why one of your assignment was to make sure that you are able to pronounce that, right? Because many times we see people, going for rotation and Streptococcus pyogenes, Enterobacteriaceae. So these are something that you really get to practice in terms of pronunciation. We already talked about the concept of colonization disease, right? Now, I'm not going to spend much time on probiotics revolution. Again, if you know the basic concept, you can pretty much determine where these industries coming from and why people are worried about that. And I think for most of the cases, I would rather wait now with these genetic techniques coming up to figure out what is normal before I declare what is it I want to replace the bacteria with. Okay. Again, ear is important because in children, they got ear infection a lot. If I was to ask you, what is the most common cause of ear infection in kids, for example, right? The most common cause is coagulase negative staphylococci. And so that also stands in terms of priority. And I think your book has done a good job. So it's going to list microorganism in terms of priority, 1, 2, 3, 4. So 1 is the one that has a higher incidence of causing problems. Again, typically, eye is colonized by coagulase negative staphylococci. But again, most of the diseases that we see with the eye are these microorganisms. Again, I would expect, I said to the students last year as well, at least 5 top bacteria you should know which are known to cause disease for that particular organ. Same goes for lower respiratory tract. These are usually very virulent bacteria. Staph pneumonia and Staph aureus, they go and cause problems. Very common cause for lower respiratory tract infections. Okay, gastrointestinal tract, as I said earlier, is filled with bacteria, right? But the most important thing that you need to know is, in terms of gastrointestinal tract, is that most of the bacteria will transiently colonize esophagus, right? And if you see, for example, esophagitis, which is inflammation of esophagus, so this is typically caused by candida, viruses, and not bacteria. So you can see very rarely bacteria because it's a transient, it's going to pass up. When we talk stomach has an acid, so these bacteria causing problem have to be acid-tolerant bacteria, right? And mostly lactic acid-producing bacteria, lactobacillus and streptococcus. And then again, cause of gastric ulcer is Helicobacter pylori. And then again, the idea for this is that many times people take too much antacids. So if you take too much antacids as a routine, oh, I've got this burning, so you're going to change the pH of your stomach. So this means you're going to give more chance to bacteria to come because now it's free because acid has been removed. So the chronic use of antacid again is a big problem. People, they do self-prescription and they want to go for that. When we go for small intestine, and these are the bacteria that you need to remember in terms of whether they are aerobes and anaerobes, and they are important in terms of causing infection. And we usually have the details when I'm going to talk about enteric bacteria. Large bacteria, as I said, if there are total bacteria 10 to 14 in the whole body, 10 to 11 are there in colon. And again, 10 to 11 bacteria per gram of feces. So you can see feces is nothing but bacteria. So people have to be very careful. As one of you just said, that you acquired from other people from going to the restroom. And I remember I usually tell this story, not a good story to tell, American Gastric Society conference, and there was a big lecture over there that everybody have to clean himself, wash his hand after visiting restroom. So they put a person over there, see how many of them, the presenters, are going to come and wash their hands. I'm not going to give you the results though. But sometimes people take it lightly, but it's serious in terms of the problem. Large intestine, you can see mostly anaerobes. Gastrourinary system, again, some of the key points I've mentioned. The chances are, as you remember yesterday, urinary bladder, because there is a flushing action avoided urine, people will not get infection in the urethra. But having said that, those bacteria which call sexual transmissible diseases, they have virulent factor. They are very good at attachment, as I said yesterday. Right? So even if you clean yourself, no matter whichever way, a single sexual act from a partner who has those STD is enough for you to give you infection because of the pathogenesis that they have. Okay. The other problem is that women will have more UTI because of the small size of urethra, and more bladder infection, kidney infection because of the small size of urethra, because of the proximity of urethra with vagina as compared to male, which have a longer urethra and the less chances for it to go up and cause problems. Again, we talked about the anterior urethra. We talked about different organisms. Some of the microbes cannot be cultivated. This is something that we already discussed at the pseudomembranous colitis, as overzealous treatment will allow Clostridium difficile to grow. And again, I'll just one or two more slides because I may not have time to talk about that, but I'm going to go a little bit detailed when I go for STDs. Dramatic influence by hormones. Most of the girls at birth will have lactobacilli, and at the time when the maternal estrogens are declined, then the flora tends to change. So that's one of the things that is important in terms of one of the STDs that I said earlier, changes with time in puberty, women using oral contraceptives, women using any other intrauterine devices or any other means of sanitation may mess up the system, and then they cause a problem over here. I'm going to let you read that. I've already picked up the important things in terms of, you can see vaginitis. Gonorrhea is a common cause of that. And there are some other bacteria as well, which should not be considered as normal and cause persistent infection. We do see very complex kind of a picture in terms of vaginal discharge in women. Cervix is not normally colonized unless somebody using vaginal douches, because it's going to push it up to the cervix. And if it does stick over there, that's those bacteria, like the commonest is chlamydia and gonorrhea, which are very important cause of inflammation of the cervix and very important cause of STIs. Okay, and I'm going to leave rest of it for you to read on your own. These are question answers, and some of the material will be repeated again when